The next topic we're looking at is called cell signaling. This is essentially where cells have to talk to each other. All right, so if these are two different cells, they will communicate with one another in a variety of different ways. So one cell can actually send out a molecule that is a signal molecule. That signal molecule will travel to the other cell. The other cell will then pick up that signal molecule. And then when it binds to the cell, it will cause something to happen. So there is a certain type of communication that happens between cells that regulates much of what cells do. Most cells won't divide or die or do almost anything unless they're told by a specific chemical signal. And those chemical signals come from other cells. And so there's a whole topic of covering the specific signals themselves, where they come from, and all these sorts of things. But what we're going to focus on really is one smaller aspect of it, not necessarily an easy or simple aspect of it, but the specific cellular response part of it. So how does a specific cell, in terms of looking at the membrane, bind the signal molecules and then relay that information into the cell? That's kind of what we're going to be focused on here. Okay. So what we have for cells are phospholipid bilayers, and we've been looking at different functions of the phospholipids. Now we're going to have a function where some of the proteins embedded in the membrane are going to have the job of being a receptor protein. And as a receptor, they're going to bind to a specific signal molecule. Now, if you watch the diffusion videos and facilitated diffusion, we saw that there were gated ion channels, and some of the gated ion channels were called signal-gated ion channels. And then those transmembrane proteins had a receptor that they could bind to open up a gate and allow molecules to diffuse through. This is very similar to this, but there's no diffusion going on here. This is just a receptor that's going to bind a specific signal and then change shape, but no diffusion happens. Okay, so it's because it's a different sort of process. So we have then certain molecules, which then are the signal molecules. What types of molecules are these signal molecules? All different types. Many amino acids or short peptides are signal molecules, but there are lipid-based signal molecules. There are a variety of different types of signal-based molecules that can bind to receptors. So it really all depends. It's incredibly huge number of them, and this is something that is still being studied. A lot of the signal molecules aren't known fully that regulate very specific cellular activities. This is also where certain cells can malfunction when there's miscommunication between the signal and the receptor. A signal binds to a different receptor and it recognizes the signal it shouldn't, or the signal can't find its particular receptor because the receptor has been altered in some way so it won't bind the signal. So the cell won't get a signal that it's supposed to receive, so it doesn't then function properly. So there are a number of diseases and disorders related to this specific aspect of cell signaling. And then some certain pharmaceuticals, what they do is they stand in for the signal molecule to bind to the receptor because the body may not be producing it properly. So that's kind of relating some of these things to some things you may know about. So a signal is very specific, for the receptor very specific for the signal. And when they bind, what's going to happen is there's going to be a change in shape on the internal side. So here, this is extracellular. The outside of the cell. Here's the cytoplasmic side. Of the cell. Okay. And so the outside of the cell, the signal binds, comes from outside, and then it changes the protein in the cytoplasm. Now that protein may do a variety of different things. There are a couple different ways in which those receptor proteins work, and we could break them down to two main categories, but there are others as well, but these are the ones that we're often more focused on. First one's called the integrated kinase activity. What that means is that this receptor, the inner part of it, is actually an enzyme. And it's turned off, essentially. And it won't be turned on until a signal binds to this part of the receptor and it activates the enzyme. And then the enzyme can do its job. So essentially the signal gets this protein to work and do a particular job inside the cell. Now, a kinase, this is something we'll get into more so later when we talk about enzymes. So all enzymes end in ASE, so it's kind of sort of a name. And a kinase is a particular type of protein that adds and removes phosphate functional groups. So the phosphate functional group, which we know from the ATP molecule, we've seen it several times now already, hopefully. It's going to take that off one molecule, add it to a different molecule. So kinase molecule, a kinase enzyme is a protein that does that particular job. And that will typically activate a protein that will carry out some function in the cell. So the signal is really saying, do this job in the cell. Make this particular protein that expresses a particular gene. But in this particular case, we don't know what it is. There could be thousands of different cellular outcomes. Regardless of all the thousands of outcomes, you have the same basic mechanism. Signal binds to a receptor. Receptor changes shape. It functions as the kinase. It activates a protein by adding a phosphate to it, and then that protein carries out a job. Okay, so it's a very more simple, the more straightforward way of doing it. However, most of the time that's not the case. Most of the time there's a slightly more complex process involved here. So I'll extend this membrane a little bit, give us a little bit more room, a little more room to draw here, and give you some other ideas of what other things can happen. So that's one type of receptor. But the type of receptor that we're going to spend most of our time focused on are what are called G protein linked receptors. Okay, this is what we're going to spend most of the time talking about. So a G protein linked receptor has a couple characteristics. Well, it's a transmembrane protein. It's also going to be what's called a 7-pass transmembrane protein. So this alpha helical segment is one pass through the membrane. A receptor that's linked to a G protein is then going to pass through the membrane 7 times. So I'm getting a different color here so you can actually see it. So you got 1, 2, 3, 4, 5, 6, 7 alpha helical segments that have been woven back and forth through the membrane. But still, essentially you have a part of the protein that's on the outside of the cell, that's the receptor part, a part of the protein that's on the inside of the cell. It's like this part here that's going to change shape and do something. But in this case, it's not the actual kinase enzyme. In this particular case, in this model, the kinase is going to be further down the road, okay, somewhere else in the membrane, and it'll have to be activated. So it's not directly activated now. This is just activating the receptor, which will have to relay the information through a few steps and then ultimately get to the kinase. So it has more steps to this particular process. Okay. So first we have, this isn't the G protein, this is the receptor. Okay. So this is what we call, again, a G protein linked receptor. Some people end up calling this the G protein and they kind of forget what we're actually talking about here. So that's a transmembrane protein. Now we're actually going to have the G protein itself. Okay, so this is, we're talking about receptor here. The G protein is a peripheral protein. So if you remember, peripheral proteins are only attached to one side of the membrane. So this would be the G protein. A G protein is this peripheral protein. It's attached to the cytoplasmic side of the membrane, and it is sort of a go-between. It's a go-between between the receptor and another protein over here, an enzyme called the effector. All right, so, and this back here. So in the original model, the receptor bound to the signal changed the shape, and then the other side of that same protein did the enzyme activity. But here I haven't even put the kinase in yet. The enzyme is going to do that particular activity. That's still further down the road. Here we have a whole bunch of intermediate steps. So signal, and now in this particular case, the signal molecule, we're also going to give it a different slightly different name, a little more specific name. It's still a signal molecule, but we're going to refer to it as the first messenger. First messenger is always extracellular. Right, so we find first messengers outside the cell. They're released by other cells, typically, and then they bind then to this receptor here. When they bind, they trigger a change in the receptor, and then they activate this particular binding site. So, before the signal attaches, the receptor cannot interact with the G protein. If they bumped into each other, nothing would happen. There'd be no type of change, there'd be no type of reaction, nothing would happen. But if the signal is attached to the receptor, then it can actually attach to the G protein. Then they can actually bind together. So now the name of this protein, this peripheral protein called the G protein. We call it the G protein because it's holding on to it a molecule of GDP. All right, so we got to say, what is GDP? So here we go. This is GTP. So if you remember, we talked about ATP. ATP and GTP are very similar molecules. Okay, so they're molecules that are energy molecules in the cell. They're triphosphates. Okay, here's our ATP. Now we have GTP down here. The GTP, if it loses a phosphate, so if one phosphate is broken off, we then have G with two phosphates, and that's called GDP. Okay, get that clear. That one's broken off. So essentially, this is a lower energy form molecule. Okay, it's kind of like ATP when it's broken down to ADP, the diphosphate. So from 3 phosphates to 2, less energy. This was at one time a 3-phosphate molecule, now it's a 2, and that's what's attached to the G protein. So we're talking about the G. G is here. I don't know why. Sometimes I ask people, like on an exam, what energy molecule does the G protein carry? And then they say ATP. I don't know why anybody would think that. G protein is the name G, GDP. It should be pretty easy to remember. So this is the molecule that is characteristic of that particular protein, and it's kind of where the energy is going to come from. Now, holding the GDP like this, that's sort of in its inactive or resting state. So when it's holding GDP, it's inactive. What's going to happen is when the signal binds to the receptor changes shape. The receptor binds to the G protein. Now it can bind to it, and it makes the G protein drop this GDP. It'll actually get rid of it. So it changes the binding site here. And the GDP goes away, but at the same time, a molecule of GTP, the more energetic, the triphosphate one, is attracted and it will bind into this site. So essentially what's happening is the signal is getting the G protein to pick up a more energetic molecule. That's kind of the idea here. Now there's more aspects of the G protein. It actually has 3 subunits, an alpha, beta, gamma subunit. Two of those subunits actually stay stuck here right next to the receptor, whilst the third, the alpha one, which has the GTP, will actually move. Okay, so it'll move down away from the receptor, and it will move to this other protein called the effector. So the effector is an enzyme. It's an enzyme that has to do some work. Right? It has to do a job, and it's going to need energy to do that job. The energy is going to come from GTP, so it's going to provide energy to this particular enzyme. So what's going to happen is the G protein, specifically this thing called the alpha subunit of the G protein, along with the GTP, the energy molecule, is going to move over here to this effector, and then it's going to do this. It's going to break off the phosphate, releasing energy, and it will become then GDP. So here, transfer of energy to this enzyme. And then the G protein will just go back to the way it was before. Then instead of GTP, it'll have lost one, so it'll just become a GDP. Get it right? It's not that it drops the GTP and then picks up a GDP. It's, the triphosphate becomes the diphosphate. The 3-phosphate one becomes the 2-phosphate one. Then eventually the 2-phosphate one is dropped, and a new 3-phosphate one is added. So more energy, lower energy. So essentially the high energy one is attached, it gives up energy and just becomes a low energy molecule. Eventually that protein, which is just sitting there with the low energy molecule, gets rid of it and picks up another high energy molecule. And then it does its job, then it just has the low energy molecule. It just goes back to waiting, and it's just sitting there by the receptor saying, should I do this again? If there's no signal, no, it won't do it. It just sits there. If a signal comes, then it'll say, yes, here, drop your GDP and pick up the high energy triphosphate and go do the job. And so the G protein is just a go-between. It goes back and forth between the receptor to the effector, receptor to the effector, and back and forth. That's all the G protein really does. And it helps by picking up a GTP, it provides the energy to this enzyme. So why does this enzyme, called the effector, even need energy? Well, it's because the job of this effector is to create something called the second messenger. So it creates the second messenger. All right, that's the job of the effector. So the effector is to create a molecule called the second messenger, which is a cytoplasmic signal. So it's going to be a different signal molecule than the one that's outside the cell. People have found through studies, if you were to take this signal molecule here, this first messenger, and you were to inject it directly into the cytoplasm, say, maybe let's just shortcut this whole thing. Let's shove it in. Nothing happens. Why? Well, because that's a specific molecule secreted outside of a cell to bind to a receptor that's located outside of a cell. There's no receptor for this molecule in the cell. There's nothing for it to attach to. Okay? This second messenger is a different molecule. It's not the same molecule that's recreated in the cell. This molecule doesn't diffuse into the cell. It's not actively transported into the cell. It always just stays outside the cell. So the first messenger is always extracellular, outside. The second messenger is always intracellular. It's cytoplasmic. It's inside the cell. Okay? If you took these second messengers and put them outside the cell, they typically don't have receptors to bind to. They don't do anything there. So each one has its own environment and where it works. So, kind of going through this process. We said this is a little more in depth than the integrated kinase activity. With integrated kinase, this receptor binds a signal. When it changes shape, it just carries out an enzyme activity. Now, we haven't even got there yet to that particular activity. All right. Here, we get the signal binding to the receptor. The receptor changes shape, binds to the G protein, which is the relay. The relay protein then gives energy to an effector who creates a second messenger. The second messenger goes on to activate the protein kinase. So the protein kinase is that enzyme, which we talked about initially, that we said is going to add or remove a phosphate for something, which essentially turns on or turns off another molecule. In this case, it's specific for a protein. It's a protein kinase, so it's going to turn on or turn off a particular protein. It's going to make it active or deactivate it. And that particular protein then has a job in the cell. It's a transcription factor. It affects the relay and reading of genetic information. It might be an enzyme that carries out some part of metabolism. It may bind to it and help that enzyme actually speed up. It may bind to it and shut down that enzyme and stop it. So, this signal is telling the cell to do something. It's saying maybe speed up your metabolism. So the way it's going to do that is relay this information, eventually activating a particular enzyme that's going to help speed up the metabolism. So it's a multiple step process. Why so many steps? Why not just do it directly? Well, here we have a lot of levels of control, right? So you have different places where these processes can be stopped, sped up, slowed down. There's a lot more regulation of these types of processes, whereas if it's just yes or no, all on, all off, it's a lot more difficult to control because cells don't typically need to be going, all out or at a dead stop. There's always something sort of in between, which is how they have to be functioning, and a process like this allows them a lot more flexibility in that. So we're going to then go over a very specific example of a second messenger system. There are two really common ones. One is called the cyclic AMP system, and the other is the IP3 inositol triphosphate system. So those are the names of two different second messengers, so two very different ones. So I'll talk about both of them briefly, but we're going to go into a little more detail in this one called the IP3 pathway, and that's going to be the end of all the material on enzymes, because it's going to come back together and put ion channels and diffusion and all sorts of things back together again with membrane structure. So that'll be the next thing we cover is a specific example of a second messenger pathway.